Good evening. Thank you for joining us for tonight's roundtable lecture, Understanding Methylation, Gene Regulation, and the MethylDetox Profile. As always, the information presented here is for your consideration and is in no way meant to replace specific consideration for your patient. Tonight's roundtable is brought to you by Cell Science Systems. Cell Science Systems is a specialty clinical laboratory that develops and performs laboratory testing in immunology and cell biology, supporting the personalized treatment and prevention of chronic disease. Presenting tonight is Dino Saleta, Rare Not. That's PhD for us in the States, from the University of Heidelberg, Germany. And joining Dr. Saleta are Amy Pizarka, RD-RDN, and Andrew Campbell, MD. If you are attending the A4M annual meeting in December, don't miss Dr. Saleta's lecture on methylation and its connection with metal intoxication and mitochondrial oxidative stress. To begin, we will turn to Amy Pizarka.
Thank you, Jason. Yes, I am a registered and licensed dietitian nutritionist and a certified clinical nutritionist. I have about 30 years of experience in this field, mostly in hospital nutrition on an outpatient basis, and as well as in private practice. And now I'm very happy to be the Director of Nutrition Services for PreviMedica. And PreviMedica is a telehealth company focused on nutrition and lifestyle. In addition to providing nutrition counseling via video, we are providing exercise and culinary guidance this way as well. And I'm working with a wonderful group of health professionals who are dedicated and passionate about helping their patients promote optimal health. And I'm thrilled to be working with them. And I'm also very excited about this webinar, the topic of this webinar, as an introduction to the MethylDetox profile, because the nutrition professional actually may be the first healthcare practitioner that a patient is consulting with who would assess folate and the status of other key nutrients. And he or she can make the connections between the nutrient status, the lab values, whether the conventional lab values or some functional testing that they're doing that may be out of the optimal range, and they can connect that with the symptoms reported by the patient. Now, judging by the questions that we received pre-webinar, I think we have a wide range of knowledge with regard to this topic. Some, I think this information is quite new, but for others, I think that others have a lot of experience with methylation and working with clients with regard to this topic. So I apologize to those of you who do have a lot of experience with this if it sounds like I'm stating the obvious. But the consideration of genetic variants is now a very important part of the nutritionist assessment, thankfully. Nutrition and how it impacts gene expression and vice versa, genes impacting nutrient needs. Knowing that information offers the nutritionist so much more information to tailor our nutrition plans to meet our patients' individual requirements. We can't change genes, SNPs in those genes, but we can sort of work around them. And not knowing the SNPs can certainly be one reason we feel we are spinning our wheels with regard to symptoms that our patients report and the lack of progress they might be experiencing. So once again, I'm excited about this topic. I'm happy to be here with Dr. Saleta and Dr. Andrew Campbell, who is the medical advisor to Cell Science Systems.
Thank you, Amy. My name is Dr. Andrew Campbell. I'm an immunologist and a toxicologist, and the best way to find out what I do is to look me up on pubmed.gov, type in Campbell AW, and the publications will light up. This methylation detox profile is so vital now in these days. We used to die of infections. The Black Plague decimated 1/3 of Europe's population in the Middle Ages. Pneumonia was the #1 cause of death in the U.S. in the early 1900s. Tuberculosis, cholera, leprosy were all familiar killers back then. Then Sir Alexander Fleming of Scotland came along, gave us penicillin, the magic pill, starting in the 1940s. Medicine started using a pill for a cure. Fortunately, we don't have the old scourges that we suffered from for so long. Unfortunately, these have been replaced by chronic health problems, cardiovascular diseases, metabolic disorders, neurological disorders, and cancers. But medicine has not shifted from focusing on using a pill to treat the symptoms of disease to preventing chronic disorders. The importance of this methylation detox profile is that it is predictive. You treat the abnormalities found in your patient with supplements and lifestyle changes. It gives you an insight into your patient's future health, and you can monitor their progress away from chronic disorders. I've had the privilege of working closely with Dr. Dino Celeda, who is now going to present his findings.
Thank you very much, Dr. Campbell. Thank you very much, Amy. I'm also very happy to be here in the United States and to work with both of you on our genetic, on our functional genetic, a little bit to my background. I got my PhD or doctorate, not how we call it, at the University of Heidelberg in Germany, actually at the Institute of Human Genetics. I did a lot of research in genetics regarding also, well, to some extent telomere length, so on and so on. After my scientific research, or let's say I started to develop genetic testing for routine diagnostics, and right now I am happy to be involved in the program of functional genetics, which we did a lot in Germany and which we are continuing to do right now here in the United States. Again, thank you very much for the invitation. And let us move on to our methylation detoxification cycle. The objectives, actually, what we want to present here right now is not getting you in all these complicated reactions, metabolic processes, biochemical pathways. We just want to give you here at the beginning as an introduction a simple pathway of how methylation or how the homocysteine and methionine balance works. For this reason, we're going to identify the most important genes involved in the methylation homocysteine cycle, their mutations, SNPs controlling this homocysteine cycle, let's say influencing, is maybe a better word for that. We are looking at the most common SNPs, very well described in literature, which actually affect the enzyme activity or the enzyme expression of the resulting gene. And then to look how the SNPs can impact homocysteine levels. And after that, we're going to also give you an overview about a personalized intervention according to the genetic test result. And here, I think from my experience, what I had so far, we are moving into functional genetics. We can give you targeted nutrition. We have also can also give for targeted nutrition. I'm sorry, I'm German. I don't find the words sometimes. Explanations. Then we also can give you some advice for targeted supplementation. And then what is actually very good, we can also monitor the progress of the treatment or the progress of the supplementation. I call it a personalized approach by measuring, especially here, the homocysteine levels. Here we are measuring homocysteine levels in other programs which we are which we developed so far, which we are going to develop. We are measuring other markers like also glutathione ratio, mitochondrial potential, and so forth. Okay. Basic stuff. What's very basic, what is a SNP? A SNP is nothing else, or also called genetic polymorphism, is a replacement of one nucleotide in the DNA. You know, the DNA is made up of 4 nucleotides: adenine, thymine, guanosine, cytosine, and the sequence of those 4 nucleotides makes a gene. What we have here right now on the left side, you see, let's say the cytosine is replaced by an adenine. And here on the right side, we have again a chronologic order of these nucleotides, and the general population is, let's say in this gene, a guanosine, and here in the polymorphic or population, and where there's polymorphic, the guanosine is replaced by thymidine. So this is actually what we call SNPs. And imagine we have here two DNA strands. You have to be aware that we have two sets of genes from father and one from the mother. That's the reason why we are all called diploid form. What does a SNP do? A SNP, sometimes it does nothing. Like we know, there are companies around who measure a lot of SNPs. We have something estimated about 500,000 SNPs in something like about 25,000 genes. Yeah? So this is a big number. So you can imagine that a lot of those SNPs are not doing any harm or not influencing any activity or expression. But yes, certain SNPs, which are described very well in literature, and we are using only those SNPs, have some influence on enzyme activity in the metabolic processes, and that's what we are looking on. The most simplest SNP and the oldest SNP we know so far is actually sickle cell anemia. In sickle cell anemia, we have exactly here one replacement of one nucleotide. And you see here on the left side, you see a red blood cell, a normal red blood cell. Here everything is in order. You see here on the left side a sickle cell blood cell resulting from the SNP. And like we all know, the sickle cell shaped red blood cell doesn't transport oxygen very well. So this is the simplest and first example of a so-called genetic polymorphism, or what we call it right now, SNP. Sorry. So what are the functional genetic test results doing? The individual functional genetic test results are shown on a level of functionality and or expression of the corresponding enzyme. Again, I have to say we are not, the genes are not first responsible for the function. These are the enzymes, and all the enzymes in our body, all the enzymes are metabolic processes. In our biochemical reactions are coded by genes. So genes code for an enzyme, and this influences our individuality. So far, like we all look a little bit different, no? Wouldn't be good that I would look like Amy.
No,
Amy looked like me. Sorry, the other way around. Okay, so we can divide that in the results of the SNPs. Remember what I said, kind of like we have one gene set from one parent, one gene set from the other parent. Homozygous positive is one of those kind of results what we're going to get. In genetic, we do everything other way around. Homozygous positive is here called minus. This means both alleles, both gene sets from both parents show a mutation. And here we can say, yes, we have a reduced functional expression of the corresponding enzyme. Heterozygous, this is plus minus. This means one allele from one parent is normal, we call it wild type, and the other allele, 50% of the genes, show the mutation. This 50% of the genes, the corresponding 50% of the enzymes, are going to have, in some cases, a reduced function or reduced expression. Then we have the situation homozygous negative. You see here again plus plus, don't get confused. I don't know who did it. I think Gregor Mendel did that, something like that, but this was about 150 years. Yeah, I think 150 years ago. So homozygous negative, both alleles from both parents show no mutation, and they are wild type. This means 100% of these genes regarding this investigated mutation sites are normal. This is a little bit explanation to go into homozygous, heterozygous, and so far positive and negative. Sometimes I see a lot of results which say homozygous, and then I ask myself, well, fine, but is it positive or negative? So we have to distinguish that a little bit.
Can I?
Yes, please, Amy.
So, just a comment. I know it's pretty complex, and we're trying to simplify it here. But the red, what we're indicating in red here, the homozygous positive, with alleles from both parents, the likelihood that there would be a decrease in enzyme activity would be higher than the heterozygous, plus minus. However, both may or may not express a decrease in the enzyme activity depending on environmental factors and lifestyle and nutritional intake. And then the one we're showing in green, the homozygous negative, this person would be less likely to have any decrease in the enzyme activity, although lifestyle and nutrition and our environmental factors are important to consider as well. Anyway.
Yeah, you're perfectly right. Kind of like you have also, when you have a normal set of genes, a lot of enzymes, especially the metalloenzymes, they need cofactors. And when they don't have this cofactor, they don't work well. Let me compare this with a car, a beautiful car, a Mercedes. And when the Mercedes doesn't have gas, it doesn't run. That's the same thing like an enzyme. For example, in mitochondria, we have a superoxide dismutase 2. It's also called manganese-dependent superoxide dismutase. Yes, this enzyme requires manganese for proper function. This means when you even are green, you need manganese for a proper function. Yeah. So this means also coming back not only to looking at the genetics, but looking at the broad spectrum kind of lifestyle, nutrition, as also, yes, sometimes targeted supplementation. Okay, what we were telling right now, targeted supplementation. How does functional genetics work? Like I said before, first a test, individual's current status. We can combine that with SNP testing and also with corresponding metabolic markers in blood. Like here, we measure the SNPs and the corresponding genes of the methylation or homocysteine methionine balance. We're going to get into that later so far, but we can also measure at the beginning also the homocysteine level. So we have a whole picture of this person, patient, call it. So with this picture, we can do a targeted intervention. This means personalized specific supplementation. Every one of us knows, or of you know, kind of what we are talking about. Folic acid, 5-methyl tetrahydrofolate in all the supplements, what are available. And also according also to the individual results of relevant SNPs and corresponding metabolic markers in blood. Monitoring again after targeted supplementation, let's call it also targeted treatment. So far, we can also monitor then the progress of this treatment by, again, metabolic markers in blood, what would be here, in our case, homocysteine.
Let me bring in the practitioner's aspect, and that is that for an organism to live, it must create new cells as fast as cells die. This requires for the body to make millions of cells every minute, relying on DNA and RNA synthesis. Mutations in the methylation pathway can cripple the ability of the body to make the building blocks needed for new DNA and RNA synthesis. And in particular, susceptible cells include bone marrow, lymphocytes, erythrocytes, and neural tissues. Remember that the nervous system is the highest concentration of RNA and has therefore the highest need for methylation.
Yeah, thank you very much, Dr. Campbell. We totally agree. Like so far, perfectly right. And Dr. Campbell and me, we have been talking so much by developing this methylation cycle. And I'm always happy. I was always very much happy. I'm still very happy for his comments and also his ideas of actually placing and targeted to certain conditions. So let's call it also diseases. Well, but let's go a little bit further. The importance of personalized targeted nutrition. Again, individual genetic SNPs, what we say, may require personalized supplementation of specific vitamins, minerals, trace elements, and or amino acids even. This is important for the glutathione synthesis, for the maintenance and optimization of metabolic processes. Fine. But an overload of supplements, and this is what we also see in a lot of cases, can impact metabolic processes and lead to side effects. Side effects, like also the newest research, newest literature shows, can influence a wide variety of functions in the organism, and yeah, some literature describe certain overload of supplements with a reduction in natural killer cell activity.
Yeah, it is. It's so true. It's so important to test. I think a lot of nutritionists, and maybe some in the audience, and myself included, we really try not to, but in some ways we end up turning to OFSA. I stole that from somebody who was speaking last night. OFSA, one size fits all. No, yeah, OSFA. Anyway, I liked it. I stole it. One size fits all. So one size fits all approach. We try not to, but sometimes we turn to that, and leery about testing, because a lot of times patients will come to us saying, you're my last resort. I've been through a whole bunch of tests as it is, and I just, you really have to help me. So dietitians and nutritionists might be leery about testing, and they might end up recommending a boatload of supplements and then just hoping for the best. And as we know, in the long run, that's not going to work. So, really, nutrition experts should not be leery about running extra tests such as this. It's so important to really know what we're looking at and to monitor.
Yeah, this is very good comment, because in Europe, actually, about 200 years ago, each doctor who was studying medicine had to study botany too, just in order to learn about the side effects of certain, still plants, certain plants, and side effects of kind of like overload with drugs, we all know. Okay. The genes, what we are looking in the homocysteine cycle, methionine homocysteine cycle, let us say a couple of words to that. Naturally, we are looking at DHFR. This is the first actually enzyme, kind of important in this whole game. We are looking at the C60770T mutation, which is very, very well described with a reduced activity, as also at the A1298C mutations, also described to influence in certain ways the homocysteine levels. This means, in other words, the methylation potential. And then methionine synthase, also coded by the gene MTR. Methionine synthase is actually the second player in the whole homocysteine methionine cycle. And we're going to show it later on a chart how this one works, but it's actually a very important gene. Here we are looking at the mutation position C351A18, and this is also described in literature with a reduced function of these enzymes. Then, as a third player in this whole game, we have COMT. Everyone knows COMT, catechol O-methyltransferases. Here we are looking also at the very well described mutation positions, G472A, or also called VAL108-158 MET, or AL52-102 tyrosine, ALA-tyrosine. This actually codes for the replacement of the amino acids, and also the mutation position G304A. And all those SNPs are actually investigated very well and described very well with reduced enzyme activity, and also highly distributed to the population.
Let me bring in
yes, please,
an
Dr.
important
Campbell.
fact here. What can make the methylation cycle go wrong? Really, there's three areas. One is polymorphism that reduce the quantity or the effectiveness of any enzyme involved in the cycle. Second, a deficiency in any of the nutrients needed by these enzymes. And then third, a reduction in gene expression, which can be caused by prescription or recreational drugs, infections, or environmental factors.
This was very, very good, Dr. Campbell, because right now you described actually the need of functional medicine, functional genetics. Not only look at pieces, look at as a whole. Kind of imagine what you said, and what we're going to also maybe also discuss later on. You have no SNPs, and you have reduced and you have elevated homocysteine levels. Well, what does it say you? Something is wrong with your nutrition.
Absolutely.
I mean, it's easy. It's easy like that. So, and this is actually our functional approach here. So last but not least, what I said, what we are looking in here, and what we are also offering is the homocysteine for measurement of homocysteine levels, well, to see the treatment progress. Okay, this is the whole cycle what we are going to talk about. And again, what I said, please, this is a very simplified presentation of the cycle. We only want to highlight those 1, 2, 3 genes we are looking at, as also the homocysteine. We are also going to show you also some ways for the glutathione synthesis, which is also connected to homocysteine. Well, and then, well, let's start. Okay, first let's start with the first step in this whole homocysteine methionine cycle, methylene tetrahydrofolate reductase, MTHFR. We all know folic acid goes over certain genes, DHFR, we don't want to go into that right now, then becomes tetrahydrofolate, and at the end it becomes 5,10-methylene tetrahydrofolate. And this is the first step which is important here in this whole cycle. 5,10-methyl tetrahydrofolate means as a template for this enzyme MTHFR to produce the so-called bioactive folic acid. And the bioactive folic acid or bioactive folate is nothing else than 5-methyl tetrahydrofolate. So, with other words, when you have a SNP or when you have a reduced enzyme activity to a SNP here, what we recommend then, bypass this SNP just in order take 5-methyl tetrahydrofolate form. And this is a very, very normal thing to do, kind of, and this is what we are always looking at, the biochemical pathways, metabolic processes. When you can do something, when you lack the enzyme activity to do something, in the give it or prescribe it as a supplement or as injection, and you Dr. Campbell know much better what we can do about it in praxis so far. Okay, 5-methyl tetrahydrofolate. Again, mutations at positions, these 2 positions we have been talking about, presence of 1 or 2 SNPs affects enzyme activity, supplementation, 5-methyl tetrahydrofolate.
So someone who does not have a SNP with an MTHFR would be more likely able to handle than folic acid from supplements and fortified foods.
Sure.
But obviously, as you said, the person with the SNP would not be able to convert that folic acid into the bioactive form. So the supplementation would be recommended, of course, because the buildup of the folic acid without the conversion to its bioactive form could contribute to a whole cascade of issues. Of course, we would still recommend that people consume the folate from folate-rich foods. You know, they're leafy greens, and most of the people in the audience, I'm sure, know the folate-rich foods. But leafy greens and the spinach and kale, legumes, asparagus, Brussels sprouts, broccoli, avocado, and so forth. So that requiring fewer steps to be converted to the 5-methyl tetrahydrofolate. But this person will likely need that supplementation, as you said.
I mean, it's easy. I mean, when I can convert that, so I don't need to supplement it. Why don't I? Well, first, let's say it in a very, very normal way. Everyone understands. I can save the money to buy me that supplement. It's very easy, and then I don't have to burden my body with unnecessary supplementation. Sometimes in Europe we say a lot of people are creating very expensive urine. Okay, so. But this 5-methyl tetrahydrofolate is not the end product of everything. It's just the first step, the very first step to enter the homocysteine methionine cycle. And here comes the player methionine synthase, which we were talking about before, or called MTR into play. The MTR so far is actually using the methyl group of 5-methyl tetrahydrofolate and donates this methyl group to vitamin B12, creating methylcobalamin, or kind of like methylcobalamin or methylated vitamin B12. Yeah, so far. And this is the first step of this enzyme, MTR. Using the methyl group, you see 5-methyl tetrahydrofolate is going to be recycled again to tetrahydrofolate, and we end up with methylcobalamin or methylated vitamin B12. In the next step, the same gene, also MTR, methionine synthase, that's actually his name, methionine synthase, donates this methyl group to homocysteine, and what the homocysteine is converted to methionine. So you see, this is also called the methylation, remethylation, or it's a lot of, it's described in literature also with a lot of names of methylation. The only thing is the methyl group is actually donated further on, step by step, in order to create methionine, and the methionine, like we know, is one of the universal donors in the body for methyl groups. Okay. Again, mutations at MTR or methionine synthase so far affect enzyme activity, and we recommend methylcobalamin. But please be sure, take methylcobalamin. There's also some other recommendations with other cobalamin or other vitamin B12. Here you really need methylcobalamin. Like the name says, you need the methyl group.
And it's important to remember those also who have an inadequate intake or an absorption of B12, even without the SNP. As people get older, the ability to absorb tends to decline. Digestion needs to be addressed. Reduced stomach acid production, maybe medications are contributing to that. And of course, vegetarians as well, the strict vegetarians, vegans, who would not be consuming any sources of B12, from their pattern of eating, because it's only coming from animals. So it's important to address that as well.
And this is a very good point what you made, as people getting older. I mean, right now you see the whole process of aging is nothing else than oxidative stress. Through this oxidative stress, we are getting more so-called not inherited SNPs which are made to our lifestyle, which we can detect, except that we sequence the DNA. And these SNPs, sure, they start also with the age affecting your enzymes activity. So when you're getting older and you have everything in fine in your SNP situation regarding the mutation sites, but your homocysteine level is still too high, then okay, I have to do something. So actually it's also very, very good monitoring tests, especially in the field of, I hate to say anti-aging, there's no anti-aging, we're all gonna die, but it's, let's say, controlled aging. It's fun.
Well, when we're aging, we're living longer.
Yeah. Okay, so this is an important step right now. And you see, this is actually here, we have here right now 2 supplements and 2 genes which are actually involved in the production of methionine from homocysteine. Okay, well, let's get us to the last step. The methionine here, like I said, is a universal methyl donor, and the methionine is converted by a gene methyltransferase to SAMe-E. Methyltransferase is actually has no such SNPs, and we can neglect here something, but S-adenosylmethionine is the major substance, also called SAMe-E, is one of the major substances, not the major substance, one of the major substances, acting as a methyl donor for degradation, or let's say for the synthesis of specific substances by COMT, catechosyl or methyltransferase. This means SAMe-E in connection with COMT are involved, let's say, in methylation of nucleic acids. This is nothing else than DNA methylation, which controls our gene regulation. In neurotransmitter regulation, here we have dopamines, the serotonin, epinephrine, norepinephrine. In xenobiotics, what are xenobiotics? Nothing else than toxic environments from the toxic substances from the environment, as also drugs. Yes, methylation is involved in phase 2 detoxification of certain drugs so far, and also in the degradation of certain or activation of certain phytochemicals. So you see, methylation processes are very, very important in the body, and this cycle here is homocysteine methionine is the major or one of the major cycles to donate methyl groups, which can be used to further on by COMT or other methylating enzymes in order to process certain methylation process in the body involved in certain fields, detoxification, and so on and so on. Yes, Dr. Campbell, please.
So let me bring in about this methylation detox profile, the certain clinical aspects.
Yes.
One is cardiovascular diseases that we mentioned earlier, and you've described this profile beautifully. So clinically, coenzyme Q10 has been used as part of the treatment for angina, heart failure, reperfusion injury after coronary bypass, cardiomyopathy, and various other disorders. The synthesis of CoQ10 in the body requires components of the methylation pathway. In particular, the last area that you mentioned, the SAMe, is where it needs adequate levels of that same SAMe that is generated in this methylation cycle. The statin drugs decrease the levels of CoQ10 in the body, so practitioners need to be aware of the methylation status of their patients taking a statin drug. Also, the relationship between elevated homocysteine levels and an increased risk for heart disease and the genetic risk associated with MTHFR C677T that was mentioned. This mutations in the methylation pathway has been recognized for some time. And in this cardiovascular area, there are two types of white blood cells that must be methylated, lymphocytes and monocytes. Low methylation in these cells lead to excessive blood clotting, resulting in thrombosis and stroke. When you step over into the neurological area, folic acid is converted to monoglutamate entities by the enzyme alpha-L-glutamyl transferase in the intestinal wall. Once absorbed, monoglutamate entities are converted to MTHF, the fat-soluble form of folate that crosses the blood-brain barrier and is utilized to facilitate the synthesis of neurotransmitters, in particular dopamine, epinephrine, and norepinephrine. In patients with methylation deficiencies, level of MTHF production is reduced, and thereby limiting the production of these neurotransmitters. And when you step into what was mentioned before, the xenobiotics at the very top there, that's our environmental toxins. We're constantly and persistently exposed to environmental toxins. There's artificial colorings, flavorings, sweeteners, and preservatives in our foods and beverages, which we test for in our laboratory in patients. The U.S. manufactures about 5 trillion pounds of chemicals annually. That's a 5 with 12 zeros. We use 2.3 trillion pounds of pesticides annually. So methylation helps clear these environmental toxins from the body. This process involves the conjugating of methyl groups to the toxin prior to removal, as well as supporting the production of glutathione. Now, as many of you already know in the audience, that glutathione is a protein involved in inflammatory control and reduction reactions, and it is important in producing and controlling leukotrienes. Glutathione is the major endogenous antioxidants produced by cells, very important, and involved in the neutralization of free radicals and reactive oxygen species. It also maintains exogenous antioxidants such as vitamin C and vitamin E in their reduced form, which is their active form. And through direct conjugation, glutathione detoxifies many of these xenobiotics or environmental chemicals and carcinogens both organic and inorganic. And this includes the heavy metals such as mercury and lead and arsenic. Every system in the body can be affected by the levels of glutathione available, especially the immune system and the nervous system, the gastrointestinal system, and the lungs. So here we see how important this cycle that we test for in the methylation detox profile can become. Lastly, I want to mention, don't want to leave cancer out of this, abnormal DNA methylation, both hypermethylation and hypomethylation is associated with cancer. In particular, low levels of white blood cell DNA methylation is associated with many types of cancers. So we can see how the importance in health of this entire cycle in this particular test.
Well, thank you very much, Dr. Campbell, especially mentioning the hypomethylation. Yes, and the hypomethylation is very well known, actually described also as global hypomethylation so far due to not right nutrition, what you have today. But I also thank you very much, Dr. Campbell, that you went into the glutathione cycle. And you see what you said right, methylation is a conjugation reaction. We have a lot of conjugation reactions for the detoxification. This is acetylation too, but also, yeah, kind of glutathione. Glutathione is nothing else than a conjugation reaction. And let me finish that up, then I'm going to show you something, how this whole cycle is interconnected with glutathione. Again, when we have here mutations in the COMT, so far, or when we have these genetic SNPs in the COMT, we actually recommend an S-adenosylmethionine or SAMe-E so far, in order, well, to, again, let's say a little bit to bypass this whole situation. So now comes the big thing, Dr. Campbell. What I'm actually also going to present in about, I think, 3 weeks. How is this whole thing connected? Homocysteine to glutathione. Well, homocysteine is nothing else than the first template also for glutathione synthesis. Homocysteine in connection with vitamin B6 produces the very important amino acid L-cysteine, and cysteine is the major component with its sulfur group, the major component of the glutathione used by the so-called glutathione S-transferases. This means transferring the sulfur group so far by conjugation in order to detoxify or to neutralize substances, toxic substances, not only toxins, but also, yes, like in mitochondria, also hydrogen peroxide. And I have right now a question asking me kind of why manganese in, yeah, was this right? Yeah, we can go into that right now because it makes the connection is very good. Why using manganese so far for the superoxide dismutase 2? Very, very easy, kind of like we have in our body so-called superoxide dismutases, which have only the task to neutralize or further process oxygen superoxide anion, superoxide radicals. And the superoxide dismutase 2, or the manganese-dependent superoxide dismutase, has the task in mitochondria to convert a superoxide anion to hydrogen peroxide, and then this hydrogen peroxide is again converted to water, yes, by the glutathione peroxidase, or also catalase. Here you see glutathione peroxidase in order to defeat or fight oxidative stress. The other superoxide dismutase are the so-called SOD1, superoxide dismutase 1. This superoxide dismutase requires copper and zinc as cofactor, is found in cells so far. And last but not least, you have the superoxide dismutase 3. This superoxide dismutase is found in arterial walls so far and is also needs as a cofactor copper and zinc. So I think I answered that. Please, for further questions, you can also email questions to us, or we're going to answer that by email, also by phone call, and so on. But anyhow, I wanted to show you here the way homocysteine, not only for methionine synthesis, but also for glutathione synthesis so far. And then you can also imagine when you have high homocysteine levels, it's bad actually for your blood or for your arteries, or let's say for your plugs. But you have to, yeah, well, you have also a high level of homocysteine for glutathione synthesis so far.
But it does serve good purposes, so a little bit is good.
Well,
Too much now.
It depends. I mean, but you see, again, here we have again the connection. I mean, don't look at one cycle. Look at the interconnection of these different metabolic cycles so far. And again, what we said, homocysteine monitoring, maintaining progress here, this cycle here for personalized treatment. And we also say in our lab reports, kind of when you do everything right here, support, give the right nutrition, give the right supplementation, and your homocysteine levels are still elevated or reduced so far, then try it with vitamin B6, cofactor of glutathione for glutathione synthesis, L-cysteine, L-glycine, and glutamic acid. With other words, let's make them this way. You did everything right here, supplementation is right, and you still have elevated homocysteine levels. Maybe something's wrong in this pathway, which have the genes CBS and CSA, cysteine beta synthase and cysteine gamma ligase, so far involved in biosynthesis of L-cysteine, or maybe further on in the whole biosynthesis of glutathione. So this would be our recommendation. Yeah.
Dr. Saleta, with regard to the monitoring of the homocysteine, what is your recommendation for how often to monitor that homocysteine?
Well, let come also to the results our lab report looks like, but I mean, Dr. Campbell can answer this much better. I'm the scientist. He's the doctor, but we would recommend 2 months, 2 or 3 months. What do you think?
I would say, depending on the patient's compliance and the severity, I would check it every 3 to 4 months.
Yeah. I mean, you see, this is also what we are. We are a lab. We know your patient only from the paper and his birthday. That's about it. So you know the person. You know his habits. You know how he eats. You know his diet. You know whether he smokes, drinks, all these little things which influence actually our controlled aging.
Also, I would mention and recommend that you do the same homocysteine test in the same laboratory every time. Don't go jumping around, because each lab has its own reference range.
Yeah, exactly. And this is very, very important. Sometimes you get, we measure the homocysteine, we get then from the doctors, but the other lab, we have different results. You see, Dr. Campbell, you're totally right. Everyone has its own reference ranges, and kind of like we would be happy that you would measure the homocysteine at us. Very nice. But when you started measuring homocysteine in another lab, keep it at that to get consistent results. Okay, good. Let's go a little bit into our, at the end, into our lab report. So far you see this is the first page of our lab report, and we also try to make it very simple. We use the whole cycle actually as a lab report. Here we have yellow COMT, like you know, yellow is 50% is normal, 50% has a mutation. In the mutation side, green is 100% negative, what we call so far what we call homozygous negative plus. Again here MTHFR, all the genes from father and mother, and okay. On the second page you're gonna get a little bit of description, background description, what to avoid when a SNP is there, what to do, kind of what you have to be aware of, for example taking antifoliates, as also here for COMT a little bit more to diet, avoiding or being aware that quercetin can also inhibit COMT activity, and I mean those little things what you have to know, whether your patient also has Parkinson treatment and so far. On the third page of the lab report, we're going to give you, like here, you remember we had in COMT yellow, this means 50% of the enzymes are normal. We're going to give you additional information, what you can do, recommendation for supplementation, as others here, in this case COMT, SAMe-E, S-adenosylmethionine, And also kind of if homocysteine levels are still elevated and everything, diet is normal, also try vitamin B6, L-cysteine, and glycine, and glutamic acid. Like we said before, homocysteine is also a template for glutathione synthesis. Last but not least, you have here homocysteine levels in the serum. So far here the person is age 48, 11.63 micromilliliter. So according to this range, what we are using here, and this is the last range, what actually which is recommended here in the US, it's still in the normal range.
So that I wanted to mention the target range that CSS has here. I know some practitioners are looking at a tighter range and look for a range of between 4 and 10 as an optimal range. So this particular guideline came from
American Heart, American Heart. We have this is 2015 recommended. You see, I mean, sure, we want to be on the sure side. I mean, you're someone you can't be attacked for, but you see, everyone knows, every practitioner knows his ranges so far, and this is the most important thing. What we are giving here is a guideline of the latest literature.
Can you comment on too low homocysteine?
Well, too low homocysteine, what we would say again, what I said in connection with the glutathione synthesis, maybe there's a detoxification going on, maybe there's really a need of glutathione for, let's say, oxidative stress, but also, again, what I'm going to maybe go a little bit into in December at A4M, also kind of in metal detoxification. I mean, the glutathione transferase M1, the specific enzyme is also T1, glutathione transferase T1, is involved in metal intoxication. This has been known for a long time. And it can be that the person is kind of like has a metal sensitivity. So far, there are different approaches to go around that, kind of with also specific monitoring testing, but also in combination with genetic testing. And here you really have also possibilities of targeted supplementation. Okay, let us move to another. I think we have 5 minutes. Yeah, let us move to the last lab report as an example. Here we choose.
This is a different patient.
This is the different patients. Yeah, exactly. Very, very good. You see here we have the whole story. Yellow, green, red. Kind of in calmed yellow, kind of 50% of the enzymes again fine. 50% show mutation. Again, empty R SNP is okay. 100% of all the enzymes are okay. In empty HFR here we have, in both positions from father and mother as a mutation, a SNP, which is affecting and really the enzyme activity so far. Again, second page, we give you all this. I say always what we say in Europe, blah, blah. But what you have to know in short, comprehensive tables, or in short, comprehensive form. So far, we give you here again the recommendation for the targeted supplementation. You see here, we have here COMT. I started a COMT, S-adenosylmethionine. On for the MTHFR, what we said before, 5-methyl tetrahydrofolate, bypassing what we say, bypassing the SNP. Again, additional information, if homocysteine levels are still elevated after targeted supplementation, take again the vitamin B6 as also the components of glutathione.
When pre-webinar, we did get a lot of questions regarding dosing.
Yeah.
And I think it's of course impossible to come up with a one-size-fits-all approach with regard to that. There are practitioners that are recommending quite a bit more than what we have here, but for our purposes, we are sticking with this. I think a good rule of thumb is to start low.
Yes.
And go slowly and follow up to monitor for side effects.
Yes, exactly. That's what
follow up within the first week.
That's what we are recommending. And Dr. Campbell and me discussed this a lot of times, what we are recommending so far. And I would, say start with, we advise start with a small dosage. And you see also the recommendation on dosage, what we did here. Some might think, oh, that's too low. But again, we have to follow certain rules so far. We have to follow certain rules. This is only what we say. You see, the lab report is nothing else than to give the practitioner an orientation so far. Again, the practitioner knows his patient, knows the person, and we are actually only giving him hints or supporting him in the treatment because, again, like I said, we don't know his background, nothing so far. So we are actually only giving orientations for a targeted, let's say, treatment or supplementation or whatever you want to call it. Okay, let's go here. Let's go to the homocysteine levels. You see this person, age 23, and remember, we have COMT yellow, MTHFR red, and here we have really elevated homocysteine levels, 34.2626 micromole milliliter. And what would you recommend, Dr. Campbell, also monitoring?
Again, this one I would monitor probably every 3 months.
Yeah. Mm-hmm. So far.
And she needs to do what she's told. She needs to follow the recommendations, and we know she isn't.
Yeah,
she's here with us.
Yeah.
Practitioners know the importance of patient compliance.
Yeah.
Play a huge role, especially in someone 23 years old.
Yeah. Well, you see, but naturally, I mean, sure, for this presentation, we picked up some kind of like very very nice cases for what we say, not so nice for the person who has the test,
right.
Extreme cases. But you can see really kind of like this impacts, and like Amy said, she's a co-worker of us, a colleague of us. Let's see how this is going to look like in about 2 or 3 months when she follows our advice. Okay.
So let us kind of get into what happens when your body is not methylating efficiently. And to cap it up, you can accumulate abnormal levels of toxic heavy metals, which are found in foods and the environment. You can develop signs of cardiovascular disease, including hypertension, cardiomyopathy, chest pain, tachycardia, etc. However, you may not have any symptoms at all until you suffer from an acute myocardial infarction also. You may develop thrombosis or stroke. You're more likely to develop neurological disorders. You'll be at an increased risk for cancer. You may be more susceptible to a wide range of inflammatory conditions, including autoimmune disorders. And you are likely to have a weakened immune system and will be unable to mount a robust immune defense against infections, which goes to show the importance of this methylation detox profile, and the application of it is vast. Many patients. I know in my practice most of the patients need this test.
Thank you, Dr. Campbell. And I think we are right now, we are over the time for a couple of seconds, no?
Well, I know we want to be really respectful of everyone's time, but there are some pre-webinar questions that came in, and I think we have them on the next slide. No? Yeah.
No. On the next slide.
Oh, okay. Actually, yes.
This
Can we go back? All right. Yeah. Let me address this. Okay. So, some of the nutrition experts in the audience may have noticed on the second page of the sample test results that there's a whole chart list of foods that are considered high in quercetin, and of course at first glance you might think, what are you kidding me? And that's how I reacted actually, because when you look at this list, it's when you just would feel uncomfortable suggesting people avoid all these things, because we're normally telling people to hit the body hard with plants, and as many vegetables as your colon can handle. And of course, a lot of these things on here are good sources of folate as well. The important thing here to recognize is that quercetin is a COMT inhibitor.
And then I can tell you, and if you remember, I showed you a pharmacological paper from 1996.
Yes.
Saying the quercetin, very well known as the COMT inhibitor.
Yes.
This was in the introduction.
Right, but nutritionists, we're suggesting quercetin all over the place. We have been, and it is in supplements as well. So, and I don't, I think the important thing here to know is that we're not saying that it's not recommended to avoid all these things. First of all, if the person has a comp SNP, just be aware of the person's consumption here, and monitor progress. And if the patient's not progressing, and you're thinking, what you want to maybe investigate further, and possibly the person might be consuming quercetin. But now, my suggestion would be, though, if you look at this whole chart, not to restrict the person with regard to all these, but maybe look towards, the left column, the first, maybe 20 foods to determine, those high quercetin foods if the person is consuming a lot of those. But it's not at all to give the message that they should be restricting these or avoiding these completely. It might be a good idea, though, for someone with a comp SNP to be avoiding quercetin in supplements.
For
However.
sure. Yeah, for sure.
So, questions on the next? Yeah, then the next. Yes, okay. So, I think red flag for practitioner to prompt the ordering of the MethylDetox profile. I think that's pretty much been covered. Dr. Campbell went over quite a few. Pretty much touches on every organ system in the body. How do you know when someone is under or over methylating? The under methylating, I think we already discussed, but over methylating.
Just order the test.
Order the test. That's what I wanted to say. I mean, under methylating, you see, we have also in Europe a lot of pediatricians who are using actually COMT. COMT is also the MethylDetox, or the whole who are using COMT, and also the other genes what we were talking about. Yes, in autistic patient, and you see, the fact is also it's very, very important, or very, very convenient, let's say it is, for pediatricians, because the genetic testing is done with saliva samples. So yeah, this is also one thing. Does someone with a SNP require lifelong supplementation from a standpoint of a supplement industry? Yes, but from our standpoint too, kind of, and that's what we are talking about, bypassing the SNP. Like we showed here, you can produce something, a specific biochemical substance in your body. Okay, try it with supplementation, and yes, the genes are never, the SNPs are never going to be changed. Never, never. Mm-hmm.
How does the standard American diet, which unfortunately is very sad, versus the paleo plan affect this? So, dietary factors overall. Well, as you know, the methylation can be affected by dietary levels of methyl donors, obviously, as we've been talking about this whole presentation, folate, B12, and other vitamins and minerals as the cofactors, and of course amino acids. So a plant-based pattern of eating, including plenty of vegetables and leafy greens and legumes, nuts and seeds, grass-fed beef and chicken, shellfish, and so forth. That would, of course, be the way to go. That would give you the nutrients necessary for the methyl donors. But with someone, the standard American diet is typically not plant-based. It's an inflammatory pattern of eating. There's not enough intake of plant-based foods and leafy greens for the most part, with the consumption of way too many packaged and processed foods overall, not providing the key nutrients needed for methylation. But also, on the other hand, with the consumption of those packaged and processed foods, the foods that the person could be consuming foods fortified with folic acid. And then, of course, if the SNP, the MTHFR, without that bioactive form of folate, could be contributing to a whole cascade of issues. So, and of course, the other thing to keep in mind is the SAD, standard American way of eating, is not only methyl deficient, but contributes to oxidative stress. With regard to the paleo plan, I know that there are a lot of versions of the paleo plan out there, but overall, with the emphasis on vegetables and protein sources from beef and chicken and fish, it can provide those much needed nutrients. But for vegetarians, the plan would be quite a challenge, of course. So it's the nutritionist's job to make sure that all the key nutrients are represented in the plan, the food and supplementation plan for each one of their patients.
Okay, let's go to the, thank you very much, Amy. Let's go to the next question. How does one decide which methylated supplement to use? Okay.
Well, we already described that
Okay.
in the lab test results. So
Yes.
when you order this test, we send the recommendations right alongside, depending on the individual's testing.
And what also has to be pointed out, what we had a lot of things, but again, like I said, you email us for further questions. 3-methylglycine was also very question, can you use that? Yes, you can use it, because 3-methylglycine is actually betaine, and in the liver the methylation of homocysteine to methionine is used with betaine. So much for that. How do I specifically use the MTHFR results on pharmacogenomic testing to help mental health patients? Here you need actually the comp. This is the most common, what we said before, what we just stated before, pediatricians so far, autism, but also to a certain disease, circumstances. We are also working with labs together, which are specialist neurotransmitter testing, and here you can also marry those two tests. You can use sometimes the neurotransmitter testing as a follow-up, as a monitoring test.
Excellent. Yeah.
Okay. I think we are it. Thank you for joining us.